Hello, and thank you everyone. My name is 𝗠cie𝗿𝗀. I'm the Product Application Support Specialist for ZEISS in the New York area, and I'll be talking about the advances in microscopy for cell biology. As well, now, different labs will have different necessities from their instruments, and the questions you ask will shape your experiments. So here at ZEISS, what drives our motivation is the needs of the researchers. And in our vast portfolio, we have a microscope for each of different situations. So we have microscopes can fill up different types of experiments, from the very small single molecule tracking all the way to model organisms. So during my talk, I'll be talking about different solutions that here at ZEISS will have to resolve different problems that biologists will encounter during their imaging quest. From diffraction breaking resolution using the 𝗬scan 2 to where I'm going to focus most of my talk, which will be how to do the new light field 4D detector, which is capable of doing instant 80 volumes a second. I'll also talk about our highest resolution microscope, the 𝗦cie𝗰𝗶𝗹𝘀, which can do up to 60 nanometer resolution. Next, I'll go over our latest LightSheet 7 microscope, which is really tailored for those long-term 3D cell imaging. In combination, we also have another LightSheet 7 that's more tailored towards large live samples as well as clear samples, more like organs. And I'll end my talk with talking about our high throughput solution and multiplexing automation solution in the AxioScan 7. Most of you might probably have heard of the 𝗬scan. This is a technology that's been part of ZEISS for a little bit over 10 years now. And for you guys, the important thing about this detector is that it can do diffraction breaking resolution. So it's not only a super resolution detector, but you can actually use it with any objective to get a more resolved image, or get a better image out of each objective. It has a really great signal to noise ratio, and due to its characteristics, we can also parallelize the acquisition. So in the next image that I'll show, we actually, in our 990 microscope and 980, we can actually subdivide the 𝗬scan detector to be able to capture 8 pixels simultaneously. And this means that for the audience is that we can image really fast in a point scanner, something that is not possible any other way. So for example, in the image you have here on the right, these are beating cilia, and right now we're imaging 143 frames per second on a point scan microscope. So if you have samples that are incredibly fast, and right now your confocal does not image fast enough to be able to acquire them, it is very likely that with the 𝗬scan with this parallelization mode, we'll be able to image those dynamic processes. As I mentioned before, the 𝗬scan can be used as a super resolution detector, which gives you around 90 nanometers XY resolution. And for everyone here, what that means is that you can get extremely small details on small molecules, as well as very fine structures for example, in this example that we have here, where we're using the 𝗬scan detector to image both actin structures as well as microtubules and also vesicles, and we can image this. In this case was a cell that just finished division. We can image this incredibly fast and with very high resolution. We recently also made improvement in the software, which allows us to deconvolve these 𝗬scan images, allowing us to extract even more resolution. So we can keep the latest for everyone here. This means that we can keep the lasers low because of this high sensitivity of this detector, which allows for low phototoxicity in your samples. Another thing that we can do is that we can use these images, or we can use the high sensitivity of the detector to be able to create 3D movies. And we can also use our 𝗶𝗳𝗀 software to track in this case, these are vesicles that have been acquired with our fastest mode of the 𝗬scan. And the point scanner, it is fast enough to image these very fast moving particles, and we can do this even in 3D. So imagine, again, as I mentioned before, if you have a sample that's incredibly fast to move and the confocal is just not scanning fast enough, it is very likely that with the 𝗬scan you'll be able to acquire the images with the necessary speed to track your molecules, and then we can use other softwares like 𝗶𝗳𝗀 to do the tracing of these molecules in the 4D environment. As I mentioned in the beginning, next I'll be talking about our new detector, like light field 4D. And with this detector, as I mentioned, we can get instant volumes, and we can do it up to 80 times a second. So just to give a quick resonance, in here we have a free swimming larva of the bat star, and these are stained with HOX, and we're obviously seeing a movie playing around that's a 3D movie. And what's new about this detector, we can actually acquire this movie in one image. So one image equals, literally as is mentioned in the page, one image, one volume. And why did we do instant volumes? And basically the reason is because life happens in 3D, and all our other methods only capture 2D at a time. So if we look at spinning disk microscopes, light sheet, 𝗬scan, and lattice light sheet, no matter how fast these technologies might be, we're always looking at the 2D plane. Whether it's using the light field 4D, we're actually capturing the entire volume that we're able to image with each objective in one go. And how are we doing this? Okay, so let's take a quick look under the hood. So the light field detector uses it's a camera-based technology, and in front of the camera, what's unique about it is that you have a 37 lens array, which, as you can see in this image, allows us to see the same sample at different angles. So as the image goes through the different lenses, each lens sends the light to a portion of the camera and allows us to see it at different angles. This allows us to actually see the full 3D view and recreate the Z stacks subsequently using Zen and 𝗬𝗂. Just to iterate again, we are able to get the instant 3D imaging because of this 37 lens array that send the light at different angles, which allows us to reconstruct the Z stacks in post-processing. What this allows us to do, images that were, or videos that were previously impossible. So in here you have the heartbeat of a zebrafish, and we're imaging at 80 volumes a second. And again, just to reiterate, this means that the bottom of the Z stack and the top of the stack are taken at the same time, so there's no misregistration in time within the beating of this heart. And then we can use a 𝗬𝗂 software to segment out the cells and track them in real time. I also like to point out the size of the volume. So this is again over 700 microns in XY and almost half a millimeter in Z. So in extremely large volume at an extremely fast speed to capture these types of events. Another real application, and this will be particularly interesting for a lot of cell biologists, is that we can image an entire organoid in one go. This means that instead of spending dozens of minutes or even hours at the microscope to image one single organoid, this image took around a couple of seconds to acquire. And we can, again, as I'm showing here, we can also use a 𝗶HS software to be able to segment out the different populations of the cells and get an accurate count of each of them. Keep in mind that for this particular organoid, we did use clear spheroids. But again, we are imaging 278 microns in Z all at once. And my last example so far for the 𝗦cie 𝗰𝗶𝗹𝘀 is this Drosophila melanogaster and observing the formation of the fat body tissue. And in this particular imaging, one image was acquired every 2 minutes for 15 hours, and the development was not interrupted, so extremely gentle imaging. I would also like to point out, just as a possible application, especially for cell biologists, is, for example, the formation of metastasis, so something that's large volume that people are looking at, that you can look at how the cells are moving within the metastasis, how they can travel and form new ones in a very easy way and gentle way. Next, I'll be talking about our microscope with the highest XY resolution, the 𝗦cie. However, the fact that it's the highest resolution doesn't mean you are compromised in terms of gentleness and speed, because it is a camera-based system, so it can be extremely gentle. We can do really fast live cell imaging, and we can actually acquire very large field of views with a very high resolution. So the key things to talk about with the 𝗦cie is that with live cells, we can get at the higher the resolution of up to 60 nanometers in XY, and if we're concentrating just into the imaging, we can actually image at 255 frames per second. However, this does not mean that we need to compromise in the field of view. As you can see in the images below, we can actually image in like entire sections of brain and entire developing organisms within a really great amount of time. It is compatible with almost all types of samples, and any fluorophore that you've been using in confocal, you will be able to bring it to the sim. And it's even a more flexible tool for imaging across different lengths and depth scales, resolutions, and magnification. As you can see, we go all the way down to molecular architectures, trafficking for sensitive samples, all the way up to, again, as I mentioned before, development organisms. Next, I'll be talking about two microscopes that complement each other. So you have the lattice light sheet 7, that's really ideal for long-term 3D live cell imaging when you need subcellular resolution. And then the light sheet 7, which is really ideal for large sample, either live or cleared. So here I have two examples, one where you have a dividing cell using the lattice light sheet 7. For reference, we get similar resolution to a confocal on XY, and actually we get more resolution in Z because the resolution on the lattice light sheet is near isotropic. So you can clearly see like X, Y, and Z look extremely close in terms of resolution. And below that you have the development of a Drosophila embryo. And I'll play that movie again. And again, this is using image analysis software 𝗶HS, where it's able to track all the cells and actually how they migrate, and actually track the subpopulations of the cells from the lineage of the parent. As I mentioned before, this is an example of a live sample, but we can also use it in combination with clear samples as well. But one thing that you might ask is what microscope do I choose, and how do I choose between these two? So here is a quick and clear example on what these microscopes do well. So they are, as I mentioned in the beginning, complementary to each other. So you have the lattice light sheet that's really good for smaller samples with a higher resolution, such as single cells, spheroids, and organoids. And when the sample starts to become very large, the Gaussian light sheet takes over as the ideal solution, such as larger embryos and mouse brain, especially clear brain. In here, I actually have a quick video of a long-term volumetric imaging using the lattice light sheet 7, and we are staining for laminin B1. And I hope you appreciate how many images we are taking, the number of total volumes, and for how long we are imaging. In this case, we're imaging a volume every minute. And as you can see, even after 7 hours, the cells continue to divide without showing signs on any phototoxicity. As you can see right now, this cell is dividing at the 6-hour time point. The last solution I'll be talking about is our AxioScan 7. It is a high throughput and a way to automatize the multiplexing that you might be able to do in your lab. With this microscope, we're able to image of up to 8 channels at a single time. And as you can see here, we do this with extremely high resolution. In this case, this is a mouse spleen fresh frozen tissue, and we have 8 different channels that we are imaging. More recently, we launched what's called the spatial biology package for the AxioScan. So we can actually there's increased speed in the multiplexing and bright field imaging with the superior sensitivity. This microscope, the AxioScan 7, can image up to 100 slides in one go, and it's what's-swappable. This means that if, for example, you need to introduce more slides while the microscope is acquiring, the microscope will just pause. The slides are inserted in these holders that you can see here on the right. And then once the doors of the microscope are closed, the microscope will just resume the image it was already taking. There is a lot of advanced automatization in the scan process, and you can do basically effortless alignment for cyclic image assay. So if you're trying to image the same location multiple times, we have a very good process to align the different images with the different stains. And part of this package, we're starting to integrate more an end-to-end solution, which combines basically everything I have here in this slide. I'm not going to go over all of it, but just to simplify, we have a slide that will have a QR code. The QR code will signify what the slide was stained with. And the stains could be acquired either from us or from other vendors. We are agnostic in that sense. And then once the slide is introduced in the AxioScan 7, the microscope will read the QR code and immediately know what workflow it needs to do. It'll image, and if the workflow includes image analysis, you'll send it further into the image analysis pipeline, and at the end, you'll get basically both the microscope acquired images as well as the image analysis with the press of as few clicks as possible. The last thing I would like to mention is that most of our software run in the same software. And what this allows us to do is actually have the microscope's image kind of talk with each other. And so here I have an example of a slide that was imaging in several different types of microscopes. So we have the overall brain that was imaging the axioscan, and then we have smaller images that were imaging on using the light field 4D to get their instant 3D view. And then we have different locations where we image using our higher resolution detector called the area-scan multiplexing, as we can see here. And all these images are referenced to each other, so we can have not just the high resolution image, but also its location on the tissue to have some information of where the actual image was acquired. So you can think that for cell biology, if you're looking at different organoids, if you're looking at different locations within a particular slide, and if you are looking for rare events, you can use a wide field microscope or the axioscan to first image the entire slide, identify those rare events, and then move it to a higher resolution microscope such as the area-scan or the 𝝻 to get like increased resolution on the regions of interest. If you like to have a demo using your own samples, always feel free to reach out to our representatives, and we'll make sure to accumulate all the needs that you might have. Thank you very much for listening. I hope you have a great day.